Welcome to the MCAT Basics Podcast, brought to you by the physicians at Med School Coach. Each week, Alex Starks breaks down high-yield MCAT topics, so you can score as high as possible on test day.
Hello, and welcome to Prospective Doctors Podcast MCAT Basics. I'm your host, Alex Starks, and our goal for today is to master more metabolism. Been following along recently with our previous discussions of metabolism, you know that we've been looking at the story of glucose once it enters our cells, and we've been viewing it through this lens of ripping apart glucose, robbing it of its electrons, putting them into this electron transport chain, which we're going to talk about today. Not only glucose during glycolysis, but also the glycolytic end product, pyruvate, and then last time we talked about acetyl-CoA and the Krebs cycle at length. And we left off discussing how we're going to continue the story of aerobic respiration, meaning when oxygen's around, and we're going to shift our focus from collecting electrons to using those electrons, in particular how to make ATP. Before we dive into the nitty-gritty, there are some things that we need to know. So glycolysis was compartmentalized to the cytoplasm, the TCA cycle was in the mitochondrial matrix, and now we're zooming into the mitochondrial membrane, the inner membrane, has all those folds that make the mitochondria so distinctive looking. And actually, a quick bio review. So remember that mitochondria have two membranes as a result of their evolutionary origins, because they used to be bacteria, or at least we think. So the folds that we think of when talking about mitochondria are the inner mitochondrial membrane. This is where all the different complexes live, or at least anchored to. And this membrane separates the inner membrane space and the matrix. And this membrane is so important because that is what's going to provide our proton gradient, which will ultimately power ATP synthase. But of course, more on that. We'll get there, more on that in a second. I also want to recap real quick where we are in terms of electron carriers, like where the electrons so far ended up. And we have NADH. We have 10 of those so far. 2 from glycolysis, 2 from the pyruvate dehydrogenase complex, and then we have 6 from the Krebs cycle. That's 3 per acetyl-CoA, but because glucose is sliced up and gives us 2 acetyl-CoA, that's why it's 3 per each one, so we have 6. We also have 2 FADH2, 1 per turn of the Krebs cycle, so 1 per acetyl-CoA. And then we've also made 4 ATP. So we had 2 net ATP in glycolysis, and then also in the TCA cycle, if you remember, we create a GTP, which is just, there's a special enzyme that swaps out that guanine for adenine. That's where we are in terms of electrons. I think we should talk about the purpose of the electron transport chain. So we'll see in a moment how electrons are passed along from these carriers through the inner mitochondrial membrane, through the different complexes. And this transfer of electron releases energy, which facilitates the transfer of protons from the matrix across the membrane into that intermembrane space. And this is going to be that proton motive force that's going to power ATP production. Now, you might be thinking, well, how does this all work? We don't really know. Science at this point knows some things. We know that the sort of the redox reaction that occurs when we're passing these electrons through the different complexes provides energy that is converted into work, which pumps protons against their gradient into that inner membrane space. But the intricacies are not really well defined yet. Maybe they will be soon, but right now they're not super well defined. Now, this transfer of electrons is very sequential. It's also very important that the transfer of electrons goes from one complex to another, and the carriers, and they're all in this very well established flow, like in order. The reason why this even works is because of a concept of compartmentalization. So from the TCA cycle, the NAD is going to be able to give its electrons to complex 1 because it's near complex 1. It's not going to be able to give it to complex 2 because that, as we talked about last time, when FADH2 has the electrons from the Krebs cycle, it's inside of complex 2. So NADH just can't even access the areas that would deposit the electrons into complex 2 because it's compartmentalized. Similarly, there's going to be an electron carrier between complex 3 and 4 that we're going to talk about, and NADH doesn't give it to that molecule because that molecule is just in a completely, geographically, it's in a different spot in the mitochondria. So that's the importance of compartmentalization. And then the last thing before we dive into the nitty-gritty that I want to talk about is the lens I think that the MCAT will reward you for looking through. I want you to think about each step of electron transfer, so from NADH to complex 1, and then further transfers from there, as being, we're passing electrons onto things that have higher reduction potentials, so more positive reduction potential. And so we are passing the electrons to things that want them more. And that's favorable, that's spontaneous. And again, like I just mentioned, that provides the power to move protons from the matrix in against their gradient into the inner membrane space. And that's going to create this potential energy that makes the ATP. So just remember, kind of apply a general chemistry, even electrochemistry. You could just pretend each of these redox events, but the electron transfer being like a little galvanic cell, if you will. And instead of using a little galvanic cell to power a light bulb or something like that, we're powering these machines to move protons. Steps of electron flow. So set the stage. The electron transport chain is literally a chain of intimately connected enzyme or enzyme complexes. All electrons will take the same route, except for when they enter into, or where I should say they enter into this whole chain. So most electrons are going to enter in from NADH at complex 1. Some electrons from FADH2 are going to enter in that complex 2. Other than that, same fate for all the electrons. Now we're going to focus on complex 1 because why not? It's the first one. And so the complex 1 is called NADH dehydrogenase. And I say it that way because you need to show up to the MCAT knowing that fact. That's very testable. So at complex 1, 2 electrons are transferred from one NADH to a coenzyme Q molecule. Now that coenzyme Q molecule is simply, we can't call it an intermediate, but it is simply an intermediate step, if you will. So it takes the electrons from NADH and it's going to help shuttle those electrons to complex 3. So that's that's the linking point, is this CoQ. Now I'm definitely oversimplifying this whole process in terms of the electron flow. What's really going on is there are a bunch of intermediate handlers or holders of these electrons. So the electrons are actually transferred multiple times from NADH through complex 1 until they reach that coenzyme Q. The first one, which I'm not convinced you need to know for the MCAT, is flavin mononucleotide, FMN. And really there are these, again, I don't think you need to know this for the MCAT, but for context's sake, iron sulfur clusters that also sort of help the electrons each time they move, of course, are going to something that has a higher reduction potential. So that's the theme. That's what you need to know. That's how you have to think about it. So if they were to ask you about on the MCAT, they would probably, I would bet money on this, they would give you a passage or more context, and your job would just be to realize, oh, hey, they're going from a lower reduction potential up into a higher reduction potential because that's how electrons spontaneously move. And the whole point of this electron flow, these redox reactions, is to produce energy for these molecular machines that are within these complexes to pump those protons from the matrix against the gradient into the inner membrane space. I'm going to continue to say that, so I apologize about the broken record, but it's important. It's also important to know that for complex 1, the electron flow through it is going to produce enough energy to pump 4 protons from the matrix into the inner membrane space. And that's per 2 electrons. Now, before we talk about how this coenzyme Q or CoQ gets the electrons from complex 1 to 3, we're going to take a sidestep and talk about entry point number 2. That's complex 2, and that is succinate dehydrogenase. You need to know that name. And you also, just as a reminder, this is physically part of the electron transport chain, and it's also physically part of where the TCA cycle occurs. So it's like it's the link there. So 2 electrons from FADH2 are transferred to our same carrier molecule, CoQ, or coenzyme Q. Now, something that really bugged the heck out of me when I was in undergrad and taking biochemistry is, why is complex 2 complex 2 if we're not going to be transferring electrons from complex 1 to complex 2? And I hate to say it, that's just the way it is. That's biology. I bring it up because I want you to remember that complex 1 electrons go to complex 3 and complex 2 electrons go to complex 3. Try not to mix that up. Complex 2 is interesting as well. It's kind of unique, and maybe one of these fun specific facts about it will help you remember the details. Complex 2 is not a transmembrane protein. I don't know, you could think about it going halfway through the membrane. So it has a portion that's in the intermembrane space of the inner membrane of the mitochondria, but it doesn't go all the way through. And the reason why I bring that up is because it doesn't pump protons. So there are no proton pumping at complex 2 when electrons are transferred from FADH2 to complex 2. So that's one thing that's very, very unique about this complex. Before we go any further about the flow of electrons, I just want to talk about coenzyme Q real quick, because it is a very, very, it's a derivative of a very, very common and important type of molecule called a quinone. Now, imagine benzene. Now take benzene and attach it at 2 different spots, the carbons, it doesn't matter where, and make a ketone there in your brain. So we have a benzene that's been modified, and at 2 carbons, it's double bonded to an oxygen. This does have consequences to how the conjugation is modeled inside the benzene ring, not important, but you need to know that a quinone is a derivative. It can get super fancy, super, super duper fancy, but it's a derivative of some kind of benzene ring with connected to 2 ketones. And the reason why they're important is because a lot of molecules in our own body take this shape, or again, are like a derivative, a fancy version of what I just described, or they're like chemotherapeutic drugs, and we kill cancer with them. So it's very possible that you would have a straight up content question about what a quinone looks like, or you could have a passage that's talking about quinones or chemotherapeutic drugs, and the association would be very helpful. So to recap, electrons from complex 1 go to CoQ, which gives them to complex 3, and complex 2 also gives electrons to a different CoQ molecule. There are a bunch of them in the membrane, and that goes to complex 3. Now, you may have flashbacks to biochemistry about this whole complicated Q cycle, because the CoQ only passes 1 electron on at a time to complex 3. So I don't emphasize that with my own students, because I don't think that it's going to be on the MCAT. Other than knowing that CoQ passes electrons 1 at a time to complex 3, I think that's really the only depth that they would potentially require. But again, with their practice materials and their content outline, all the analyzing I've done, I don't think it's going to be on there. So we're not going to talk about it. But what we are going to talk about is complex 3. And this complex is called cytochrome C reductase. And yes, you got to know that. Now, cytochrome C reductase, or complex 3, is going to accept 2 electrons from CoQ. Now, it is, again, 1 electron at a time, but you can bookkeep it as 2 electrons. And that's going to go through complex 3 and be given to cytochrome C, which is the carrier molecule between complexes 3 and 4. You can think of that CoQ and cytochrome C as in principle the same thing. But the unique thing for cytochrome C is it is a 1 electron carrier. So whereas coenzyme Q can drop off only 1 electron to complex 3, it still carries 2 at a time. But cytochrome C doesn't have the room for extra electrons. It can only carry the 1. And for every 2 electrons that go from CoQ to cytochrome C through complex 3, and it's kind of the mediator, we're going to pump more protons. So we're going to pump 4 protons per 2 electrons that travels through complex 3. And it's the same logic as complex 1 proton pumping. We have these favorable spontaneous electron transfers from a species that have a lower reduction potential to a species that has a higher reduction potential, and that generates power for the complex. In whatever mysterious way it pumps it, is able to move 4 protons from the matrix against its gradient into the inner membrane space. Since we're on the topic of cytochrome C, I want to just point out that it's very likely you will see a practice passage or an actual passage on your MCAT that cytochrome C is doing a very, very different job, and that would be the job of apoptosis. And I kind of feel sorry for cytochrome C, because not only is it the oddball that can only handle moving 1 electron at a time, it also has the job of leaving the mitochondria and telling other parts of the cell that, hey guys, it's time to kill ourselves. It's kind of a gruesome messenger, a job that it has to play. So I always feel bad about cytochrome C. It really just has like the worst luck. But that also illustrates a point here, is that we're learning content, and that's really the focus of this episode, at least, is to get the facts out there that you're going to need to know. But you have to be very mentally flexible. So if you see another molecule that you know is traditionally in a certain process or does a certain job, but you have a passage that is suggesting it does something different, well, just go with it. And you'll be able to apply other foundational content knowledge you have to get you to the correct answers, but just don't be intimidated, because it's a very normal type of passage to see. So cytochrome C takes its 1 electron, does 1 at a time, and it's going to go and give that 1 electron to complex 4, which is called cytochrome C oxidase. This should really help with the complex numbers, because cytochrome C reductase, complex 3, reduces cytochrome C, and then complex 4 oxidizes it. Not too complicated. Now, I just realized I didn't mention something I want you to know that I think is important. So we did talk about how CoQ is, which transfers from complex 1 and 2 to 3, that lives inside of that inner membrane. But cytochrome C actually exists in the inner membrane space. So it exists above or outside of that membrane. It has a functional group that kind of keeps it tethered to the inner membrane, but most of the molecule exists in the inner membrane space, where all the protons are. So now that the cytochrome C has traveled over and is transferring electrons onto complex 4, I want you to visualize it as if the electrons are moving from like an above position through this protein complex. It can look like a blob in your visualization, all the way down back into the matrix. And when 2 electrons come into the matrix, they're going to be paired with protons there to make water, because there's oxygen that's sitting at the very bottom of complex 4 just waiting to get its electrons. So that's why we call oxygen as the terminal electron acceptor. And this is a very important concept, because if there's no oxygen, well, it can't accept those electrons at the very end. And so this whole process, the electron transport chain, the TCA cycle, all of that starts to back up. You can think of it as a traffic jam or something like that. Essentially, metabolism, like aerobic metabolism, just stops working if there's no oxygen. It's also important to think about what else you can say, or what other conclusions you can come to, given that oxygen is the terminal electron acceptor. I'm actually going to ask a couple of those questions at the end, but it'll be a very good sort of example of what the MCAT is going to want you to think about when you're given that kind of fact. The last thing I'll say about complex 4 in this step is that because protons in the matrix have to combine with the electrons coming from cytochrome C in order to form water out of elemental oxygen, O2, it only pumps half of the normal amount of protons from the matrix into the intermembrane space. So 2 protons pumped, not 4 like complex 1 and 3 do. This means that 10 total protons were pumped from the matrix into the intermembrane space due to the spontaneous redox reactions from electron transfer in between all the different species or molecules in this chain. Now, something to also be careful about is that you don't mix up the protons and the electrons. I almost said electrons instead of protons about a dozen times already, and you have to be very careful, self-monitor that you're not mixing those up, because that's an easy trap, easy point to lose. The next thing we need to talk about is what's called the proton motive force. Now, this proton motive force is created by all that pumping, because what we've done is we've made the intermembrane space very acidic compared to the matrix, so a lot of protons in the intermembrane space, not so many in the matrix. That's an electrochemical gradient, so we can use that for something. We can use it to power something that's non-spontaneous. And if you haven't already guessed, the non-spontaneous thing that we're going to power is ATP synthase, which is going to make the ATP. This part of the whole process is called oxidative phosphorylation. So let's dive in, ATP synthase. So this is a motor. It's actually very elegant. If you have some free time, maybe after the MCAT, would be a really cool topic just to go and learn more about. It's very interesting because it is a motor. And so this motor, we use this proton motive force from that chemical gradient, and we make this ATP, right? So physically, the ATP synthase has two subunits. We call them F0 and F1. These work together, again, kind of in a motor fashion, through a process called chemiosmotic coupling. So what that means is we are coupling, like we're pairing. The coupling and pairing is another way to say this. The exergonic process occurring in F0 to an endergonic process occurring in F1. So that might seem a little bit abstract, so let's talk about the subunit. So F0 subunit is an ion channel. So this allows the protons, this is like a way for the protons in the intermembrane space to make it back into the matrix where they want to go. So that's spontaneous. We're harnessing that spontaneous influx of protons back into the matrix to do something. And that's very exergonic. Provides enough energy for F1 to take an ADP molecule, which is actually quite stable, and then attach that third phosphate, the gamma phosphate, onto this ADP to make ATP. ATP, not stable. And we've talked about this a little bit, and it's not stable, and we know that because then when we hydrolyze it, when we cleave it to make ADP, we get a ton of free energy. So that's a clue right there that it's not very stable. It's because we get a lot of energy when we destroy it. Now, the level of detail about ATP synthase in particular, not the electron transport chain and what we've talked about in general today, it's kind of up in the air. And the reason, let's unpack that. So the reason why I say that is because the AAMC content outline, the AAMC practice materials really doesn't suggest that you need to know many facts about it at all. Other than that, it makes ATP. It has something to do with this proton motive force. But I have heard rumors, I'm not going to say specifics, but I have heard rumors that there have been some nitty-gritty questions on them, on people's actual MCATs. And the rumors are very few, so it's not like everybody's coming, or even close to that, coming out of their test and saying, hey, Alex, I heard this thing, or I saw this thing. So that's the reason why I'm not going to talk about the nitty-gritty. It's not all that complicated. It's just a lot. It's just a lot of detail. But if you are well past your goal score, this would be like an area to spend a little bit of time, just so in case you're the unlucky one that gets an ATP synthase question, you're prepared. But again, not high yield, not something you have to worry about too much. I want to talk about the totals of ATP and whatnot, kind of like the numbers, because that's a much more of a common question. So 1 NADH provides the energy to create 2.5 ATP. Of course, that, I remember in biochemistry when I took it, they told me 3. But for the MCAT, it seems like they want to go with 2.5. So the 10 NADH molecules we've obtained from glycolysis And the TC up until now give us energy to make about 25 ATP. Similarly, FADH2 equates to around 1.5 ATP. So we've had 2 FADH2s that are produced from 1 glucose. And that gives us 3 ATP. So we're about 28 there. But then also glycolysis makes 2 ATP net. So that's about 30 so far, 30 ATP. And then the last 2 ATP we have to think about are the GTPs that are created in the TCA cycle. Those are just readily converted into ATP. So that, at the end of the day, 1 glucose molecule through aerobic respiration, so like when oxygen is around, gives us 32 ATP per glucose. So something that I think is really cool is, let's say O2 is not around. They're like, oh, we're just like wasting glucose. Like we're getting 2 ATP net for anaerobic glucose and 32 for aerobic. Why do we even do anaerobic? It's not as bleak of a situation as you might think, though. So we talked about in the glycolysis session about producing pyruvate, which can be converted into lactic acid. And we're about to embark into details that are not on the MCAT. But I think conceptually this can be very helpful. So that lactic acid can go into the bloodstream, get picked up by, I think the red blood cells pick it up. I could be wrong though. Anyways, the point is the lactic acid makes its way to the liver. And the liver has a set of enzymes that can do this thing called the Cori cycle. Don't need to know the Cori cycle. But it turns the lactate or lactic acid back into pyruvate. So that pyruvate can actually make it into a cell that would do aerobic respiration. So we do get that much ATP for most glucoses that still initially undergo that anaerobic metabolism. So a couple things I want to talk about before we kind of wrap up is what happens if we break the electron transport chain. Now, conceptually, this has shown up in so many AAMC passages and questions. That's why I think it's so important. You probably can't really predict how exactly you're going to see it, like an actual test, but we're going to talk about how to think about it. So let's consider cyanide. We've all heard of cyanide, I'm sure. So this is a type of inhibitor, like a direct inhibitor for the electron transport chain. And what happens, so cyanide is going to, if you ingest it, it's going to make its way to the inner membrane space in the mitochondria. It's going to find complex 4. We've talked a bunch about how in each of these complexes, the electrons go through multiple steps. So there are like multiple molecules or species within the complexes that sort of pass the electrons along, like intermediates. And there's an iron. There's an iron 1 in complex 4, in particular, that the cyanide is able to find and access, and it binds to that iron. So that iron is no longer able to accept the electrons and then pass them on to whoever, maybe it's another iron, I'm not sure, on to whatever else is supposed to accept the electrons, ultimately to get those electrons down to oxygen. So we're breaking the complex at that point. So we need to think about everything up until that moment, that step in complex 4, isn't broken. It should be running normally. But everything downstream from that is broken. So what that means, it's not just the oxygen accepting the electrons, it's also going to interfere with the ability of ATP synthase to make ATP. So all those James Bond movies or Mission Impossible, I don't know, those movies where there's cyanide poisoning or you hear about it, all that's happening is that this molecule fits that iron ion better than this electron that's going to be passed to it. So, and that ruins everything, kills you. There are a bunch of other inhibitors, some that I guess attack ATP synthase directly. I think it's rotenone. I could be wrong. You might want to fact check me. There are molecules out there that affect different aspects or steps of the electron transport chain, but at the end of the day, it's the same thing. We are blocking it, like truly blocking it at that step of the chain, and so everything just sort of falls apart. The other type of inhibitor isn't, I guess, a complete or suicide type inhibitor that cyanide's like. It doesn't just end the electron transport chain's function. This one is called an uncoupling agent. And there are multiple out there, but one that is very famous is called 2,4-dinitrophenyl. Now, one of the quiz questions at the end, I'm going to ask you about the structure, but we'll get there. Anyways, 2,4-dinitrophenyl, which I'll call DNP from now on, was a molecule, or it is a molecule, that a lot of factories and manufacturers used in the early part of the 20th century. Heck, they might still use it, I don't know. That's kind of when it was discovered, and it was in like paint dyes, kind of like the yellow paint that you put paint curves. And I remember reading something about it being in dynamite or something, but anyways, I digress. So this molecule, people that were around it and using the chemicals and materials that had this molecule in there were losing a bunch of weight. And of course, a magic diet pill is going to attract attention. So what happened is doctors, scientists, whoever sort of isolated this compound and put it into a pill. And it was a quote-unquote obesity cure. And it worked really well. Very, very well. Except some people started dying. That took too much. Like most things. A little bit's okay, a lot's probably a bad idea. So they were dying in a very interesting way. They were overheating. They were dying of hyperthermia. Like too hot, temperature unfolds the enzymes, and if you don't have enzymes and proteins to carry out all the jobs that cells and your body needs to carry out, you die. So they discontinued it. But I will say, when I was in college, I had a roommate who actually got some of this. And this is a terrible idea. Do not do this. He got this because he wanted to lose a little bit of weight before some trip or something. And he started taking it. He didn't take too much or anything like that. I was careful. And I just thought it was so funny because he would just be sweating and sweating and sweating. And it was always just so hot and miserable at taking this, quote unquote miracle diet pill, which is very, very dangerous. And so I started looking into this compound because I was not tempted, but curious. And what happens is this molecule, this DNP, embeds itself in the intermembrane space. I don't know why that membrane is its favorite to go to, but that's where it goes. And it allows the protons in the intermembrane space to come back into the matrix without going through ATP synthase. Usually, like we talked about earlier, the protons need to come through that ATP synthase in order to get back to the matrix. That's that proton motive force. But this is a shortcut. And so what happens is the electrochemical gradient between the intermembrane space and the matrix goes away. It starts to go away. And what that does, so you're making less ATP, right? And so what that does is it turns everything into overdrive. So it's hey, we're not making enough ATP. Let's get more substrate, and usually in body fat, if there's no glucose around, bring that in. We'll run it through the TCA cycle. We'll get the electrons and we'll keep it going. To make enough ATP for the cell's needs. And every time that we have one of those electron transfers within the ETC, some of that energy is released as heat. So that's one of the major ways that we heat our bodies actually, are just that the normal transfer of electrons in the ETC. But this molecule was making it so that all of metabolism just had to work so much harder and faster in order to keep up the amount of ATP to keep the cells alive and whatnot. That's why my roommate was so hot and sweaty all the time, is because of that. I still laugh about it to this day because he was so miserable. But yeah, so don't do that. It's just a nice analogy to kind of think about how we could mess with metabolism in a way and have some really real consequences. All right, so that's what I've got for the ETC for today. I want to talk about where we're going to go from here. From here, we're going to talk about fatty acid metabolism. In particular, we're going to talk about beta oxidation. We're going to avoid fatty acid synthesis for now, which is not a very high yield topic, but I want to get through all the catabolic parts of metabolism before we start talking about the anabolic processes. And the theme for the fatty acid, the beta oxidation, is going to be looking at an alternate source of acetyl-CoA, because when beta oxidation breaks down, it breaks down to acetyl-CoA. But of course, we are going to do some questions. Right, so the first one, as promised, I want you to think about what the structure of 2,4-dinitrophenol looks like. Give you a second to think about that. All right, so I want you to picture a benzene ring. It's 2,4-dinitro, so carbon 2 on the benzene ring and carbon 4 on the benzene ring have a nitro group, which is an NO2 group. The carbon 1 has to be a hydroxyl because it's a phenol. So it's 2,4-dinitro, those are your substituents, and the base molecule is the phenol, which is just a hydroxyl attached to a benzene. All right, my next question is, and you can bring in outside knowledge to this if you want, or other metabolic pathways, etc., but what molecule in the mitochondria has the highest, most positive reduction potential? The answer is O2, because that is the final electron acceptor. So by definition, if it's the final electron acceptor, that means it wants the electrons the most, and so must have the highest reduction potential. If cytochrome C, for example, had or a poison, not something like cyanide, you ingest something that has a higher reduction potential than oxygen and also makes its way into the matrix or into the electron transport chain, well, that's going to win the electrons, and that would, of course, destroy the electron transport chain because you're not getting the electrons over to oxygen. All right, last question, kind of a softball. What physical or chemical feature of DNP, 2,4-dinitrophenol, allows it to reside within the inner membrane of mitochondria? All right, so that's what I planned for today. If you have comments, suggestions, questions even that you'd like clarification on, feel free to email me at alex@medschoolcoach.com and we'll get those taken care of. But until next time, when we talk about beta oxidation, best of luck with MCAT study. See you all later.
Each episode of MCAT Basics is brought to you by Med School Coach. To access Med School Coach services, including MCAT tutoring and medical school admissions advising, visit our website at medschoolcoach.com. Good luck as you prepare for the MCAT, and we hope you tune in again next time.